Hi, good day everyone. So this is my thesis entitled Shell Morphometric Variation of the Freshwater Snail, Melanoides tuberculata, across the 7 lakes of San Pablo City, Laguna, Philippines. So freshwater gastropods are a group of invertebrate mollusks that are very diverse in nature since there are approximately 4,000 species identified worldwide. And these are composed of 3 clades, which are Eritomorpha, Cynogastropoda, and Heterobranchia. Freshwater gastropods are important animals since they serve a lot of ecological uses. First, they are microherbivores, which means that they consume primary producers such as bacterial films, algae, and agricultural crops. They also serve as prey for a lot of molluscivorous animals such as crabs and fish and even humans. They are a good model for studying phenotypic plasticity. Now, what is phenotypic plasticity? Phenotypic plasticity is the ability of an organism to modify or change its physical or genetic traits to adapt to a certain environment. But the question is how and why? Well, first, it's because they are slow moving. And since they're slow moving, they're exposed longer to environmental selective pressures. Secondly, they're sensitive to changes, and this prompts them to change their physical or genetic traits to adapt to that certain environment. Thirdly, they are good environmental bioindicators as they could reflect the current status of an environment where they reside in. And lastly, they have a high fecundity, which means that they could reproduce several of their offspring, thus allowing them to pass on their modified traits onto the next generations. So now let me discuss Melanoides tuberculata, which was discovered by Buehler back in 1774, the snail that I used and studied for my thesis. So it is an invasive cynogasteropod snail under family Thyaridae. And since it's invasive, it can colonize a wide range of aquatic ecosystems. It's also resistant to harsh environmental conditions, such as eutrophication and desiccation. And since it is invasive and resistant, it is cosmopolitan distribution. It can be detected in almost every freshwater habitat. And you may ask me, why is it that I chose Melanoides tuberculata, knowing that it is resistant to changes, knowing that snails are supposed to be sensitive? Well, they're still sensitive, but they still persist in these kinds of environment, but they do still express phenotypic plasticity in the form of morphs, based on the study of Pointer and colleagues in 1993, which is why I used them as the primary snail of my study. Now let's move on to the Mar Lakes of San Pablo City, Laguna, and Luzon. They are geographically located in the Bacolod Corridor, and they're formed through phreatomagmatic eruption of Mount Banao San Cristobal, where big circular depressions were formed and were eventually filled by rainwater over time, thus creating these lakes. So these 7 lakes are Mohicap, Yambo, Pandin, Calibato, Palakpakin, Bunot, and Sampaloc. And the main thing about these lakes is that they highly operate on aquaculture ever since its first introduction in Bunot Lake, which was passed on eventually to the other lakes for the commercial production of fish, such as tilapia. Secondly, they also operate on ecotourism as observed in the lakes Yambo, Pandin, and Sampaloc lakes. And lastly, domestic activities, since there are settlements that are also found nearby each lake where these people reside in. The main problem, though, is that these lakes suffer from water quality deterioration, given that their activities are unregulated, therefore resulting in the decline of their waters. And based on the study of Mendoza and colleagues in 2019, they have actually discovered that most of the parameters of these lakes didn't conform to the internationally set standards for healthy lakes, especially in terms of water temperature, DO, and pH, based on these graphs. And another study by Demzon and colleagues in 2018 also conducted a survey in Lake Sampaloc, Palakpakin, and Pandin for the presence of organic pollutants. And from there, they discovered that Sampaloc had the most organic pollutants, followed by Palakpakan and Banding, and these usually come in the form of pharmaceutical compounds, surfactants, organophosphates, artificial sweeteners, and a whole lot more of organic pollutants, given that domestic activities also operate in these lakes due to the presence of human settlements. Moving on, let me now discuss the biodiversity of the 7 lakes. Apparently, it is very limited since I've only found 3 studies that conducted biodiversity studies, such as Ichthyofaunal Diversity Service, or fish, zooplankton and phytoplankton compositions in Lake Bonot, and macrogastropod diversity, which is apparently only conducted in Lake Sampaloc. But surprisingly, one of the snails that they have actually sampled was Melanotis tuberculata, and this was a great indication that I should also use Melanotis tuberculata since they could also be found in Lake Sampaloc. And it is a huge proof that it could also be found in the other lakes. So after reading a lot of studies about Melanotis tuberculata and the 7 lakes of San Pablo, I've established a research gap wherein there is still no study that tackled the relationship between the water quality of the 7 lakes and the morphology of the freshwater gastropods, including the variation of gastropod morphology across the lakes. So from there, I established my objectives with the help of my thesis advisor. Generally, it determined the possible relationship between the shell morphometric patterns of M. tuberculata and the water quality of the 7 Mar lakes of San Pablo City, Laguna. It specifically determined the shell linear and geometric shape character changes of M. tuberculata, and lastly, analyzed the relationship of the morphometrics of M. tuberculata shells with the water quality of the 7 lakes. So my study only focused on the morphometrics of the M. tuberculata shells as exposed in field conditions, and it did not cover the variation in terms of its visceral mass and tissues and the effect of predators on the plasticity of their shells. So we established the 7 lakes as our study sites for the collection of these snails and the measurement of the environmental variables. We divided each lake by 10 hectares to establish the sampling sites in the littoral zone specifically for the collection of the snails and measurement of the environmental variables. So for the measurement of the environmental variables, a Garmin 12 handheld global positioning system was used for the geographic coordinates, and the multi-parameter YSA professional plus water meter was used for the measurement of these physical-chemical variables. As for the field sampling, we conducted it during these months in the wet and dry seasons. And as I've mentioned, they were distributed in the littoral zones, every 10 hectares. Snails were stored in sealed bags and conical tubes with absolute ethanol for preservation and for other studies that were conducted by my other colleagues at the time. Upon collection of these snails, they were subjected right to linear morphometrics, where its shell height, shell width, body whorl height, and aperture length and aperture width were measured using a digital caliper or ImageJ software, and these were exported to the past version 3.26 software to run principal component analysis. The same snails were also used for geometric morphometrics, where digital photographs had to be taken first with a DSLR camera for landmarks to be added on each photo using the TPS package, and they were exported to MorphoJ version 1.07 for this analysis to be run. Generalized linear mixed model was then applied using RNR Studio, where the following environmental variables were set as fixed factors. The sites were set as the random factor, and the size and shape based on linear and geometric morphometrics were set as the response factor using these packages and importance values. So now moving on to the results of my study, I've actually observed based on their averages of their linear characters that Lake Bonot and Lake Sampaloc had the most distinct average sizes. This showed that Lake Bonot had the smallest shell linear character sizes, whereas Lake Sampaloc had the biggest shell linear character sizes, while the rest of the lakes had intermediate sizes in between Lake Bonot and Sampaloc. So to further prove this, a character loadings and a scatter plot was then generated based on the principal component analysis in PAS 3.26. And from here, you would actually see, as demarcated by the green convex hull, that Lake Sampaloc indeed had the largest linear sizes. And while Lake Bunot had the smallest linear sizes as compared to the rest of the lakes, which were intermediate of these two. Moving on, we now have geometric morphometrics, where we generated a canonical variate analysis graph just to show if there are overlaps. And there are actually, in fact, a lot of overlaps. But upon generating a transformation of Whorf grids and morphing diagrams, we actually saw that there was a distinct difference wherein the shells exposed in the shells found in the CV1 or canonical variate 1 had wider but shorter body whorls and smaller apertures. And the shells from the CV2 had slender and slightly taller body whorls and wider apertures. So from there, after conducting linear geometric morphometrics, we aligned them from each other using principal component analysis of one, which is the size from linear morphometrics compared to the shape, which is the geometric morphometrics of the snails, shell snails, which was the relative warp one. And this is also aligned to the environmental variables that were set as the fixed factors from the generalized linear mixed models from R&R's studio. And from here, you can see that temperature had the most significant effect on the size and shape of the melanoidous shells. And you can see here that there's a distinct separation difference between Lake Sampaloc, Yambo, Calibato, and Pandin from lakes Punot, Mohicap, and Palakpakin. And from there I was able to determine that Lake Punot, like I mentioned, had the smallest linear character sizes, but they also had narrower and taller body whorls and wider apertures. Sampaloc, on the other hand, had the biggest shell linear character sizes, but it also had based on its shape, the shorter and wider body whorls and smaller apertures. The rest of the lakes, however, had intermediate linear sizes between the two, but then Lakes Calibato, Yambo, Pandin had the same shape as Lake Sampaloc, whereas the rest had the same shape as Lake Punot. So though temperature was found to be the environmental variable that had the most significant effect, we can't exactly use that as a primary basis for explaining the shape and size changes of the shape of the snails, since there are a lot of variables that can actually contribute to those variation and changes. And one is the presence of pollutants due to aquaculture, based on the study of De Chavez and De Lara and Falconer and De Chavez, which proved that heavy metals can in fact induce developmental abnormalities, both in terms of their shell and visceral mass. The presence of organic compounds as well, such as tributyltin, as I've mentioned. These lakes also highly operated domestic activities, which is why they harbor a lot of organic pollutants. And though I didn't exactly include predators as part of my study, predators in fact do also have a lot of effect as well in the shape shifts of snails based on these studies. So here I have my summary conclusion, which only highlighted again the things that I've said in the previous slides. And as acknowledgements, I'd like to thank these following organizations: the National Research Council of the Philippines, University of the Philippines, DOST, and the Seven Lamps for making my thesis possible. And that is my study and presentation for today. Thank you.